Welcome everyone to our seminar series. We have our second international speaker for the year. One of the positives of the virtual series is our footprint in the world for our guests has expanded, so we've taken advantage of it. We're very happy to have Fritjof Helmchen today, visiting virtually from Switzerland. So Fritjof studied physics in Germany at the University of Heidelberg, and then he did his PhD at the Max Planck Institute in one of the kind of great environments in neuroscience with the Sakmann Group, Nobel Prize winner Bert Sakmann, where many great people were doing great work at the time, including Fritjof, really studying the details of synaptic transmission and the calcium dependence of these things, and really pushing much of the technology that Fritjof and others have pushed the use of in systems neuroscience today. And he, after his work with the Sakmann Group, where this was his PhD work, I believe, he then did his postdoctoral work at another great environment, the Bell Labs environment, where David Tank, Winfried Denk, Karl Swoboda, others were really at the forefront of pushing two-photon and multi-photon imaging of neural activity. So these two really great environments were seminal for the work that Fritjof has set up in his own group. And he's continued to really push the use of the technology to have insights about neural computations. And he's focused mostly on the barrel cortex, the somatosensory system, and how neural circuits are mediating information processing, and now moving into how they're using this information during behavior. Since 2005, he's been in the Brain Research Institute at Zurich, where he's the co-director. That's where he's joining us from today, and where he'll be telling us about his work. So thank you for joining us, Fritjof.
Thank you very much, Bilal. Thank you so much for the invitation. I will now try to share the screen. Yeah, welcome everyone. Thanks for the invitation. As I said, I'm calling in here from Zurich from my home office, and I'm pleased today to present to you some recent work we did on wide field calcium imaging. And the work I'm going to present to you is mainly the work from two group members, from Ariel Gilad, a former postdoc who has moved on already to Israel, and Yazia Galero Salas, a former PhD student who is now a postdoc in the lab. And my group has over the years really become very interested in examining the relationship between what you can call brain dynamics and body dynamics, or body dynamics being equal to like behavior. So we moved from the more cellular and local circuit scale now also to the large scale of even looking at dynamics throughout many interconnected brain regions, and in particular during behavioral paradigms. And one of the themes that I will touch upon today is that, this is a complicated, intricate relationship, because not only controls brain dynamics behavior, the motor actions, but also motor actions behavior itself will reflect back onto and also modify and modulate brain dynamics. So it's a very complicated loop, and this will be a recurring theme throughout my talk. Now, Bilal already mentioned we've been working a lot on the barrel cortex, that's shown here, and the task we designed also then relate to tactile perception and tactile discrimination. Oops, and I should I'm sorry. Maybe I can. Is this better? I'm sorry, there's the sound. I hope it's good like this. Mm-hmm. So a few years back, Jerry Chen, when he was a postdoc in my lab, he designed this tactile texture discrimination task, which is shown here in action on the upper right. A mouse, head fixed under a 2-photon microscope in this case, has to discriminate two types or multiple types of sandpaper textures, rough or smooth sandpapers that are presented to its whiskers on one side of the snout. We designed this as a very basic, classic go-no-go task, where one of the textures is sort of the go textures upon which the mouse is supposed to lick and receive a water reward, and another texture is a no-go texture where the mouse is supposed not to lick, to refrain from licking, and then wait for the next trial. And obviously there's the two other trial types. It may miss the correct texture, which would be a mistrial, or it may lick on the no-go texture, so which would be then a false alarm trial. So a classic go-no-go task. So this is in the meantime been very much established in the lab, and we have used it in sort of slight variations in different studies. And we also set up then to look at more complex tasks based on this. One I will touch upon in this talk is the delayed response task, where we added a delay phase after the presentation of the texture. So after the texture is withdrawn, the mouse still has to wait for a certain time, several seconds, until it is allowed to report its decision, and then can report by licking again by the action. Another paradigm we've been recently used, but I will not have time to go into it, I decided, is also reversal learning, where actually after the animal has become an expert in the discrimination, we reverse the value of the textures, reversing go and no-go texture, and then look at what happens upon this reversal learning when the animal have to deal with this rule switch. What I will almost exclusively show today is the simple optical technique of wide field calcium imaging, which really can make very nice use of the available transgenic mouse lines that express highly sensitive calcium indicator, even specifically, for example, as here in a particular cortical layer. So we've used in all of actually what I'm going to show you from wide field imaging was done on these layer 2-3 mice, where GCAM6F is expressed in a triple transgenic paradigm here, mostly and densely in layer 2-3. So this is something just to keep in mind. And we've also confirmed, like many others, that what we measure here in terms of fluorescence changes is really related to the underlying action potential patterns that occur in these layer 2-3 neurons. And I will show one slide later, only one, where I will also show validation experiments using 2-photon microscopy. But otherwise, this technique is really a simple fluorescence microscope with a camera. And the beauty is that also in terms of preparation, we do not even have to open the skull, but we can work with an intact skull preparation like this, where we just take away the skin and clean the skull and then stabilize it with transparent dental cement, and then can literally image through this skull for weeks and months. And this obviously also gives the opportunity, the great opportunity, to look longitudinally in the same animal, for example, what happens during learning of a task. And then we just use an Orca flash camera, like here, a CCD camera, to record the fluorescence images. This is what it looks like, sort of in terms of like raw signals, if you like, here color-coded, like the GCAM6 fluorescence. And then in terms of pre-processing, what we can do is we are interested in obviously finding out where activity happens and relating it to the brain anatomy, to the mesoscale regions as defined, for example, and specifically here by the Allen Reference Atlas. And the way we do it is that we also use anesthesia sessions where we apply sensory stimuli from different modalities, whisker stimulus, a visual stimulus, auditory stimulus, forepaw, hind paw. And this gives us these sort of maps, these functional maps, sensory evoked maps, which we then can use as anchor points together with bregma and sort of the anatomical landmarks to register our movies to the Allen Brain Atlas. That's sort of shown here. And what we look at here is essentially the dorsal view on the cortex, where we can detect about 25 regions, which are listed here on the left, including the sensory regions, barocortex here, number 18, visual cortex, in green here the auditory, primary and secondary areas, in pink here the association areas that play a major role in my talk, in particular these PPC, posterior parietal cortex areas 13 and 14, we'll come to that later, and the motor areas like here in front. Okay, so the questions equipped with this sort of nowadays rather really simple technology is that we really wanted to ask, well, first of all, where do we see task-related cortical activity, also in the different phases of the task, and in particular also in the delay phase. We'll come to that later, which essentially is a short-term memory phase where the animals have to hold certain information in memory. But I will also touch upon, we had a recent study, and I will briefly touch upon, we're also very much interested in what changes in cortical activity occur during learning and how sort of the learning process relates to the changes in brain dynamics. I think this is a fascinating field where we are just now having the tools at hand to really investigate it. Then, as I said, in particular, the focus will be in my talk on the short-term memory phase when you have the delayed response paradigm. And there we will in particular also ask, because this was a salient finding of ARIA, what the influence of the motor behavior of the mouse is, sort of how the dynamics we see in these cortical areas depends also on the movement behavior of the mouse and how whether these are really influenced by behavior. And we do see very strong dependence on behavior which led then to the second and last question here, in how far this generalizes across sensory modalities, not only maybe for the tactile modality for whisker tasks, but also for auditory tasks, and I will come to that in the last part of my talk. Overall, we always characterize the learning curves of the animal by calculating the performance according to a D prime value here, and usually set here the threshold in a dashed line where we consider that the animal has sort of become an expert at a D prime of 1.5, like about 80% correct trials. And you can see these are individual animals, that the animals obviously are different in their learning curves. Some are fast learners, some are slow learners. So this is the typical variability that we also see across animals. Okay, so let's jump into it. So let's start with the most simple, just go, no-go task, and first look at the more primary and secondary areas. And in this case, obviously, this is a tactile whisker detection or whisker discrimination task. So let's look at barocortex and the surrounding secondary somatosensory cortex S2, and also here between visual cortex and barocortex, part of the PPC, which in this case is the region RL, the rostrolateral region as part of PPC. And this is work from Ariel a few years ago, where he started to look at this. We give first an auditory tone that is a cue indicating the trial start, and then the texture is moved in and then stays in for 2 seconds. And the first touch with the whiskers to the texture actually often happens a little bit before the texture stops, because the animals obviously can whisk. They're free to whisk, and they can sort of whisk against the incoming texture, and thereby having an early touch, which may occur even like half a second or so before the texture stops here. Now, the one thing that we noticed in expert animals, and that's very consistent, is in expert animals you see that in HIT trials, in this case the P100 texture, which is the rough texture, was the GO texture, and in the HIT trials the barocortex signal here is larger compared to the correct rejection trials, which is the smooth texture here. Here below is another animal where we trained the animal for the smooth texture as GO texture. So here the P1200 texture was the GO texture. And again, also here we see sort of that then this in these HIT trials, the signal is larger. So it's not related to the roughness of the texture, but it's related to the HIT, the go versus no-go association, which we also had shown before, like in Jerry Chen's work. And this is also shown down here in the, and it's not only true for S1, but also for S2 and the PPC area, that in expert mice, we do see sort of an increased, enhanced response, stimulus response for the HIT trials compared to the correct rejection trials, which also means that all these three areas are then able to discriminate the textures or the HIT versus correct rejection trials rather early, like after half a second. You see how the two curves diverge in all cases, and the red bar here indicates when this is sort of significantly different. This obviously at some point also has to arise during learning. So if you just compare the same signals in barocortex for a naive animal and an expert animal, then you can see in the naive animal the go and no-go texture actually evoke very similar wide field signals here in the barocortex, average signals. But in the expert animal, that's where you then see that this discrimination power is enhanced, comes up, and then the HIT trials for the go textures show the enhanced signal. And obviously also later here, when then the licking, the reward collection happens, you also see a marked difference, because here obviously they collect reward for the HIT trials and they don't lick for the correct rejection trials. We also analyzed this discrimination power using an ROC analysis, classic analysis, as many of you probably know, where you look at the true positive rates versus false positive rates, and then you can define the area under the curve as a nice measure of discrimination power here. We can even create the maps here, and you can see that barocortex here has the highest discrimination power in the expert mice. One thing that I want to mention, and you see here, is obviously then again the variability and also in the maps. And one factor that immediately becomes obvious if you do these types of experiments is that the activity maps can look very different. And one aspect is the activity of the animal. And we call it here active and passive. I'll explain it later, but I want to mention it here already. Active just means that the animal is very active in terms of movements during the sensation, during the sampling of the sensory stimulus, while passive means that it would be in a more still mode and passive mode and just passively receive the stimulus. And when the animal is moving, you then will see a lot of extra activity in many cortical areas, particularly also in frontal motor related cortical areas, or here like hind limb, forelimb areas. So very strong signals, rather widely distributed, whereas in the passive case you see a more confined activation actually of barocortex, sort of the primary areas. But I'll come back to that later. Before that, I want to first show a little bit what happens during learning, what we found for learning. For learning, as I showed you, from the naive to the expert animal, obviously things are changing and there seems to be an enhancement in barocortex. But how is this coming about and what else is happening? So to do that, Arya really measured longitudinally in a whole cohort of mice from the beginning, when the animal had not even seen the task, when they were naive, throughout many trials, thousands of trials, until they had become experts. And that gave us the opportunity to really look a bit more longitudinally what is happening. And we focused on these like three time windows that I already briefly introduced, related to the cue initially, then sort of the intermittent pre-stimulus period, and then this stimulation period that starts a little earlier than the texture stop. Now, in a naive animal, this is data from one example mouse. In the naive phase, you do see the cue evoked like auditory cortex activation, also here often related to it like hind limb activation. Then you see sort of meandering activity in the pre-stimulus phase, and then you clearly see also the whisker touch evoked activity in barocortex. Now, when this animal has become expert, yeah, then you still see a similar pattern, if you like, here for the cue. You see the strengthened signal for the barocortex, but what is particularly salient is also the sort of intermittent phase of the pre-stimulus period, where you now see a strong signal in this PPC area RL. And that was sort of one salient finding we saw and were interested to look further into it. The nice thing is for any of these regions, you can now not only look on the trial time scale what is happening. This is the example for barocortex, because we have the entire time course of the many trials throughout learning. So this is on the one hand shown here. This would be the trial time course going to the right. And then going from top to bottom is the learning time course, the much slower learning time course. These experiments were done, this is the simple task version, so these experiments were done over the time course of something like a week, like 5 to 11 days, during which the animals learned the task. And then you can look at the delta F over F signals either on the trial time course, which is shown here, and you can see again the strengthening of the stimulus evoked signal, or you can then look on the learning time scale across all these trials. And here you can see the enhancement of the barocortex signal in the stimulation window during learning. Now, in RL, for example, then you also see the strong increase here with learning. But then there's also peculiarly other areas, for example, other, if you like, association areas here more medially, the posteromedial area, PM, also kind of a secondary visual area, and also rostrodorsal retrosplenial cortex. They actually also show a decrease of activity. That was something we were also wondering about. And when you look at the time course, it was also interesting to see that this decrease in, and this is the Q-related decrease then here in PM and RD, actually happens before learning, right? As you can see here. So here we try to align things to the learning threshold or indicate the learning threshold. And you can see that the enhancement in barocortex and RL kind of happen with learning. But these decreases here actually happen before learning, which we found interesting because it indicates, oh, there's something that's even happening before the learning really starts, if you like, or really happens. And this was consistent for like all the animals and led us to look into it a little further by actually fitting to all the different areas, all the 25 areas, fitting a model that included like this suppression in maybe an early phase, and then this enhancement in a late phase, sort of surrounding the learning threshold when the animals reach D prime of 1.5. And this gave us a chance to define something like a modulation index, where either the areas would be more suppressed or they would be more enhanced, showing more this late enhancement. And if you now look at the signals for these three different phases, in the Q phase, there's overall more like suppression with the learning than here, which may indicate that the animals have gotten familiar with the task. They know the try start cue, so they really just perform the task, and it's not like a very sort of salient cue then to them anymore. But what we found very interesting is that in the pre-stimulus phase, in this transition phase, there's a very large sort of suppression in the association areas, particularly, except for RL, right? Because RL sort of sticks out as highly enhanced together also with barocortex, the two highlighted here. So, which means in a way that upon learning, it seems that there's this specific enhancement of the task-relevant areas, if you like, which we see here. And then in the later phase, when it comes to the stimulus detection, then we see also a lot of further activity in further sensory and also motor areas because the animal starts to lick. So from this, and you can look in the paper for more details. I won't go into more details, but from this we just get this very rough idea and model that there might be two phases of learning. One would be a more like non-specific suppression of actually many areas. That's in a way what we kind of see, which may just reflect the prerequisite for learning. So the way I think about it is that it may just indicate that the animals get in the right state and the right mode of to be ready for learning. For example, that they would stop just indiscriminately licking and actually not paying attention and enter like more an attentive mode where they really are now able to figure out what the task actually means and what they have to do. And this would go then hand in hand with the sort of specific enhancement of the task-related activity in the task-related regions, like here RL and then barocortex, in a way setting up the correct sequence of events from the cue to the maybe even prediction of the touch, then to the actual touch that is then this sort of the task relevant reward predicting stimulus. And we think this is maybe a general idea, or it would be interesting to follow up if sort of there's a more global suppression. This is shown here for, in particular, in the association areas, a global suppression followed by a specific enhancement of the task-relevant pathways. If this could be sort of something that is also happening in other similar tasks. Good. So as first conclusions, I've just shown you now that the S1, S2, and RL show this early discrimination power in expert mice, and that during learning we find that a particularly strong refinement happens in the association areas, in this case for the texture discrimination task, in particular enhancing RL. This now I move to the second part where I really want to focus on the short-term memory, which brings us to the a little bit more complex task version, which is the delayed response version. And this is something that already Jerry and then Ariel in my lab have set up, which requires a lot more training of the mice. So this can take weeks to really train the mice so that they not only discriminate the go and no-go texture, but they also wait with their reporting until there's a response queue, which basically sets up this delay period, which can be several seconds and usually several seconds long, where obviously the animals now have to hold at least some information in short-term memory, either about the texture type, if it was the rough or the coarse texture, if it was the go or no-go texture, or they could, in this go no-go task, they could also just in a way keep the information of their decision if they want to go, like looking forward, if they want to go, or if they do not want to go, if they want to sort of refrain from licking. And the mice can actually learn this, as is shown down here. These are just two example mice, and also one of them trained on the P100, the rough texture, one of them trained on the P1200, the smooth texture, as go tries. And you can see that then once they have reached expert level, they will perform actually very nicely. We had one mouse that even could do delays up to 10 seconds or so. Sometimes they will lick too early. We call these early licks, and we have actually not further analyzed these early licks, just excluded them from the analysis. Now we come back to this other aspect I already mentioned briefly, which is that what Ariel then detected is that the animals, one really has to look what the animals are doing during the trials. because they can behave very differently in terms of movement and movement activity. And we then defined really two main types of activity which relate to the movements during the sensation, during the touch. This is the same mouse. On the left where the movie is running, you can see what we call an active touch, an active touch, where the texture comes in and you can see the mouse not only whisks sort of against the incoming texture, but it also moves the limbs and moves the back, and the whole body is in action, if you like. Now I hope that the right one will also run. I think I have to maybe switch back my pointer. How do I get back? Because the mouse is not that passive. It's not moving, not at all. Like this. Okay, now I can run it. I hope you can also see it. Now this is a passive trial. It's the same mouse, so I also want to highlight that the mice can do both. And you can see what we refer to as a passive touch is when the animal really is still and not moving during the touch. Later, afterwards, obviously it starts moving also in preparation of the licking action. Both of these are hit tries and correct hit tries. So the animal can perform correctly using both active and passive touches. As I already showed you, the activity during this sensation, during these touches will be very different. In the active tries, there will be a lot of activity, including frontal motor areas, limb areas, and so on and so forth, while in the passive it will be more confined. And for us, a key question was now, how does such a behavior-dependent cortical activity impact also on the short-term memory phase, which comes afterwards, which is then the delay phase where information has to be held in memory. And to do this, Ari also had to quantify the body movements. Nowadays, probably one can do this even better with like a pose estimation software, and we're using also Deep Lab Cut now a lot in the lab. But here he just analyzed it in a rather simple way using the cameras, not only of the whiskers, but of the monitoring the body movements, and then just looking at the frame by frame correlation between the subsequent frames, so where he could actually detect movements in particular areas relating to the forelimb and to the neck and the back of the animal. Then you get curves like this here, and you can set a threshold, and then you can binarize it, and this would be the animal is moving, the animal is not moving, and this would be an active trial where there's movement during the sensation period, and this would be a passive trial where the mouse is quiet during the sensation. And I want to really highlight that when we talk about active and passive, we only mean active and passive during the touch itself. In the later delay phase that we are going to analyze, the animals are not moving. At least we are only looking at the delay phase periods where the animal is not moving. So everything I'm going to show you is related to the sort of previous activity in the sensation period. And trials like this that are noisy, where there's also movement during the delay immediately after the sensation, we actually discarded. So these are the active, these are the passive trials. Mice are different. Some mice are using more like the active strategy. For example, this mouse here, mouse number 5, is very active. This is now movement probability calculated across many trials. And you can see this is very active almost always during the sensation. This is the period where it's then still, and then it starts moving again in preparation for the hit trials, the licking. This is sort of a more mixed animal, which sometimes is active, but often also not so much. Again, here in the hit trials, you see then the preparatory and licking action. And this is more also like a passive animal that rarely shows activity during the sensation and performs the task in a more passive way. Now, the salient finding, and this is the sort of one of the, I think, still key findings of Ariel, is that he recognized that there's completely different delay activity patterns for these two types of trials, the active and the passive trials. This is shown here for one example mouse. So now we're looking at the delay phase, yeah, as I said. So we're looking at the phase when the touch has happened, it's over, the texture is withdrawn, the animal has either moved a lot, being active, or it has been sitting quietly passive. And now we look at the activity that's in this delay phase before it's allowed to lick and report its decision. And what he found is that in the active trials, we see persistent activity in the mediofrontal areas, which we call here M2, but it could also include the anterior cingulate cortex, because we are here very much in the midline where the cortex is obviously diving down. And in the passive trials, because this is the same mouse but different trial types, in the passive trials there was almost an opposite activity pattern where we saw persistent activity in the posterolateral areas here, almost even lateral to visual cortex. I'll come to that in a moment. But very far away. I mean, this is almost centimeter away, right? But very consistent. So these are on the left single trial examples from one mouse when it was active, when it was passive, and here the averages. And you can see less activity in the correct rejection, maybe a little bit still here, but overall less activity in the correct rejection trials. Now this was very consistent across mice. And as I said, there's different types of mice, individual personalities. Some are more active. We quantified this in terms of this activeness, how many active tries or what fraction of active tries they use. And some are more passive, like these mice, 7, 8, and 4. And in the active mice, we consistently saw the mediofrontal persistent delay activity, while in the passive mice, we saw consistently this posterolateral activation spot here in all these mice, right? And we also quantified this. If you group the animals according to their main activity patterns, you can see here the orange is the sort of increase, enhancement in the M2, what we call here M2 for simplicity area in the active trials, and in this posterolateral areas P for the passive trials, right? I should maybe, I mean the M2 area or like mediofrontal activation is maybe not so surprising, at least maybe was not so surprising to us, because obviously people have reported persistent activity like in the medial prefrontal cortex, also during working memory. But the posterolateral persistent activity was a bit more surprising to us. So we looked a little more into it, and still we have to look more into it. First of all, what areas are these? One thing that we did is we compared it to the mapping of the visual cortex in like all these 4 animals. And you can see that consistently the spot where the delay map showed a hot spot was kind of just posterolateral to V1 in all these 4 animals, right? So this essentially is, if you look at anatomical previous work, like from Burkhalter, you can see this relates to the areas here, area P, POR, or LM, like classically maybe secondary visual cortex areas, but we think that our experiments also clearly show that they do not only have a visual function, but are probably more like integrative associative areas. So we have not fully pinpointed exactly how the local circuits there are. I think this would be very interesting in the future. If you look at the Allen Brain Atlas, it's also like this spot here, which again is the POR, LM, or what they call posterolateral area here, corresponding to P. Now here's my 2-photon slide, because we wanted to really check if this is also true on the cellular level. So Ariel performed a couple of 2-photon calcium imaging experiments where he, in these layer 2-3 GCAM6 mice, where he opened either a posterior window or implanted a posterior window, or like a window above like the M2 area in the frontal window. And And then he could really resolve, obviously, then the individual layer 2-3 neurons and measure their activity during the task. And what he found was, in a way, very confirming that indeed he found that in the active trials, like for this, these mice here, he saw delay activity. These are now just pooled cellular activities was high, stayed high in M2 or in this mediofrontal area, which also could include ACC, but not in the passive trials. While when he measured 2-photon in the posterolateral area, he could not see this delay activity in the active trials, but he did see a persistent delay activity in the passive trials. So it was very, on that global level, without going into further details, it was at least confirmative for our wide field data. Now, one thing that came up as a question is, what information is then actually held in these two areas, like M2 and P, if we call them like this for simplicity? And one thing you can do, we did, is you can also look at the error trials, which is kind of interesting, because a part of the hit and correct rejection trials, you also have the false alarms and misses, and you can ask, if you look at the delay activity patterns, you can ask which look more similar. And what we found is that for the active trials, the hit looked very similar to the false alarms, and the misses looked very similar to the correct rejection, which means that they looked very similar for the same action. So basically, they seem to be more motor related. Yeah? While for the passive trials, like in this mouse here, we found that actually the hit trials look more similar to the miss trials, and the false alarm look more similar to the correct rejection trials, which means they look more similar for the same stimulus, because obviously false alarm and correct rejection trials are the same stimulus and miss and hit, which indicates to us that they are indeed more sensory related. Yeah. And if we now, from these types of comparisons, similarity comparison, calculate an index, a sort of sensory motor index, we can actually see that indeed in the mediofrontal areas there is more motor related information sort of represented, encoded, which in a way obviously makes sense. And in this posterolateral areas there is more sensory related information encoded, which at least would be consistent with the idea that in the posterolateral areas maybe you really have information about what texture has been presented, and you use that information for then in the end licking or not licking. While in the other case, in the active trials here, things have shifted already to M2, and essentially the decision has already been made for like go or not, or go or no go, and it's already in the premotor cortex, and it's only waiting to release the brake. Yeah, this would also then suggest that we, or it came up as a question, whether this is then really also causally linked to the performance. So if that's true, that this delay activity is really essential for the behavior, the performance, then we should be able to perturb it. So we used optogenetics to prove this. The first thing we did is we used VGAT generorhodopsin mice, so mice that express generorhodopsin in the inhibitory neurons, a classic way to inhibit sort of local regions across the cortex on the level really of, let's say, 0.5 millimeter or so. And we only targeted then the light illumination to the delay period here. And consistently, when we shone light on both of the areas, M2 and P, we consistently decreased the performance level significantly, even below here our expert level. And Ariel then also performed nice experiments using, instead of the VGAT mice, RHT viral expression that he targeted particularly to the areas that were mostly activated. For example, in the passive mouse, the posterolateral area. And then if you block now, if you shine light only on the posterolateral areas, we did see a decrease in performance in this mouse. It did not change its activeness. And the nice thing was that you did see the decrease in performance only for the passive tries, but not for the active tries, which means we presume that during the active tries, actually the delay activity was in the mediofrontal area. So that then in this case, this perturbation did not matter so much. I noticed that I can switch maybe back to the laser pointer. That's nice. Now here's another animal that also is interesting, where Ariel tried to perturb the mediofrontal area in an active animal, where most of the trials were active. In this case, there was a slight decrease in performance, but not that dramatic, still significant. And again, also here it was nice to see that the perturbation caused performance decreases for active trials, but not passive trials. But what we noticed is also that this animal seems to be smart because it apparently changed its behavior also. So while it was very active in the baseline during the perturbation sessions, it started to become more passive, as if it realized that somehow the active strategy didn't work so well anymore, and switched sort of its behavior. And actually, when we stopped the perturbation, it switched back to becoming more active. So that's something one has to be careful about, because obviously this can confound the interpretation. But we actually found this a very intriguing result. Good. So the idea from these experiments from Ariel are really that I should maybe watch at some point. Yeah, I'm way too slow. I guess I have to accelerate, but we're coming to the last part now. But to summarize briefly, for Ariel's idea is that really from the sensation which activates S1, S2, and PPC, you will have a divergent routing dependent on the activity during the sensation. When the animal is very active in terms of movements and in engaging in the touch, it seems that for the delay period, the activity is shifted and stays there in this mediofrontal regions. If it's passive, it's shifted the other direction to the posterolateral regions, and from there somehow then converges on the correct response, because they can perform correctly in both these tri types. We also think this is consistent with anatomy, and propose that PPC, like the area RL, could be a routing area, because it has been shown previously, like from Burkhalter, that these PPC areas like RL, they have projections to both, like the mediofrontal areas, M2, and also to these posterior areas. So we think from there signals could be then routed either way, and this is what we're interested to look into further. Yeah, so to summarize this, we identified really these highly distinct delay activity patterns, and also we found that they're really contingent on the motor behavior, on whether the animals are active or passive during the sensation. Now, the last questions I want to now show the results from Yazir is we wondered if this is now something peculiar or particular to the tactile task, or if this could be also true for other sensory modalities. And that's why Yazir, who I show here, he extended the study, including now a very similar, actually pretty much parallel designed auditory task. So down here, this is the usual texture discrimination task with delayed response that I've just shown you. Up here is the auditory task, the analog task, where we have a light cue, not an auditory tone anymore as a cue, but a light tone, a light brief blink of an LED. And then the animal has to discriminate two different frequency tones as a stimulus. And then again, it has to wait, and then there's sort of a visual response cue. Now, I think the great work of Yazir was in particular that he even managed to train 4 mice sequentially in these two tasks, which is kind of heroic, because training a mouse in a delay task is very laborious and can take here several months. And here he even then managed to retrain them on the other task after these months, so the entire experiment takes at least like almost half a year or so. But it's obviously very useful because now we were able also to compare the activation patterns even in the same brain for the different tasks. Then also in the auditory task, we analyzed obviously active and passive tries. We had to stratify the tries. We did it very similar to ARIA. And interestingly, also in the auditory task, which was kind of a little bit unexpected, we saw that there are animals that are using even active, active tries, even though you might suspect that they might not need to be active for the auditory task. But we still saw the entire spectrum of them being either still or even active. And also interestingly, we found that if you then look at the 4 animals that were trained in both tasks, that they also could actually change their activeness. Yeah. So, for example, this very active mouse in the auditory task became much more passive or more passive, but there's also a mouse here which was passive in the auditory task and became then more active. We can discuss maybe what we think why this is. Now, comparing first the sensation phase before we come to the delay. In the sensation phase, obviously, we're now speaking about 2 different modalities: auditory stimulation and whisker stimulation, and this is reflected in the first activation patterns. And obviously, we see auditory cortex, particularly this secondary area, auditory dorsal, anterior dorsal area, activated with a tone stimulus, and we see barocortex activated with a whisker stimulus. Now, a huge difference that we identified that we find interesting is in the PPC areas. Because remember, for the whisker stimulation, we always saw this very large engagement and specific engagement of the RL region, the lateral PPC. Now, for auditory cortex, the peak of activity here is in the medial PPC, which is this area A. And this was very consistent across animals. So here you can see all these dashed lines are auditory task, the area A, and all these are in the barocortex task, in the whisker task, which then have the activation spot in RL. So we think this demonstrates that there's different subdivisions of PPC engaged in these 2 different tasks. Now, interestingly, we also then used optogenetics to in a way prove this. So Yazir did again experiments on the VGAT mice, but this time now he targeted the perturbation, the inhibition, targeted to either the A areas of PPC or the RL areas of PPC. Yeah, and basically during this sensation, during the stimulus presentation, he tried to suppress activity in these areas. What he found is that in the auditory task, suppressing A had a large effect on performance and a big drop in performance, but not shining light on RL, and vice versa in the tactile task, where you basically saw an effect when you inhibited suppressed activity in RL, but not in A. So very consistent with our idea that these seem to be sort of important nodes through which signal flows that are important for solving the task. Now we come to the delay. So again, just to mention, also for the auditory task, I already said so, we also have active tries. Just to remind you, also for the active tries, we will have much more larger, widespread activation, including motor areas, also here in the auditory task. But how about the delay activity patterns? Now, here's an example of a mouse which was trained in both tasks, first auditory and tactile. And here it was rather active in the auditory task and rather passive in the tactile task, but still it will do both types of tries in such one session. This is just one session. So we look into 2 sessions, and then we will have a few examples of passive tries and active tries in the auditory task, and a few examples of passive tries and active tries in the tactile task. And what does it look like? Well, actually it looks fairly similar. Meaning that for active tries, also in the auditory task, we see mediofrontal persistent activity, and in the passive trials we see posterolateral association areas being activated. In a way, rather similar pattern, or at least a roughly similar pattern to what we see in the texture discrimination task, which is here shown and very consistent with all what I've shown you from Ariel's experiments. This is the overview from several mice where you see that it's really like these almost opposing poles where we see this short-term memory related activation. I would not obviously dare to say anything more specific, because you see a little bit that there might be a shift in the activity here in M2 for auditory and tactile activation. Obviously there can be fine differences, and one should look into probably best with 2-photon microscopy. This one I will skip this slide because I think I should also wrap up, but I also want to make one sort of methodological side remark. So one thing obviously that comes up as a question, well, is this all consistent with anatomy and the connectivity? We looked into the brain atlas, Allen brain atlas, and indeed you find obviously that there's a higher connectivity among the primary and secondary auditory areas with the area subdivision in PPC-A, and RL is sort of shows a higher activity actually with S1 barrel cortex and S2. So this in a way is making sense in the findings that we have, what the patterns look like during the sensation period. Now for the delay periods, we were particularly interested how the PPC areas in RL and A relate to M2 and the PLA areas here. So Yazir did injected retrograde AEVs, either expressing a TD tomato or an EGFP. And then we looked into sort of the cells that project to either M2 or project to the PLA. And what all these data indicated here, and this was done with light sheet microscopy, which I will still want to explain in a second, the main finding is that there was very little overlap of the labeling in these areas RL and A, indicating to us, this is something I think will need to be further checked and tested, but indicating to us that there might be different subpopulations in these PPC areas that project to either more frontal regions or to more posterior regions. And this would also be obviously one way to explain how this routing can happen between either more shifting the activity frontally or posteriorly. So before I end with one slide, I just want to highlight, it's more like an advertisement, I want to open a bracket, a short remark on the light sheet microscopy that we also used here for the anatomy. Just for people who are interested, and I know many of you are maybe also technically interested. And maybe you've seen that another student in my group, Fabian Vogt, who is now a postdoc also, that he actually came up with a very nice open source light sheet microscope. This is what we call the Mesospim initiative. So who's interested, please go to the website, mesospim.org. It's an open source microscope in the sense that all the details how to build the microscope and how to run it are there freely available. And in the meantime, there's already more than 11 of these microscopes have been built around the world. And it's working really nicely. The idea is that it's a light sheet microscope for large clear tissue volumes. Here we show it for an entire mouse brain, but we even have done entire mice. So you can go to really large volumes, and it has certain advantages compared to commercial instruments. For example, it has a very nice modus where you can achieve isotropic resolution, single cell resolution across the entire mouse brain. This is just one coronal section, and this is sort of the bad view, the XZ view. For the microscopists, they know immediately what I'm talking about. It's the bad view because here actually the light sheet, as you know, is not completely flat, but has a waist and then becomes broader, so that at the edges here of such a huge field of view of almost like 20 millimeter or so, that you get all these obviously cigars instead of cells. But one trick you can use is what people have done before also is actually scanned light sheet microscopy, where you scan the waste of the light sheet together with the rolling shutter of your camera. And the effect is really amazing, because it brings you from this basically to this here. And you will never want to switch back to the old modus, because now you really have three-dimensional isotropic resolution on the order of a few microns, so that you can resolve individual cells throughout the entire mouse brain. You can even see here dendrites. So this is one of the advantages. The other is that it's also built to be very rapid. It can take whole mouse brains in just a few minutes. It's very easy sample mounting, can also handle very large tissue blocks, as I said. And it also has is working with manageable data sizes, because it doesn't have the highest resolution, but as you can see, the nice thing, it has this isotropic resolution. And another advantage, it's compatible with essentially all different clearing techniques. So if you're interested, check it out. If you're lucky and you have sparse labeling like this here in the cerebellum, just as a fun example, this was just by chance finding in one mouse line that it has this beautiful, bright, sparse expression in Purkinje neurons, but you can see the capability of the light sheet microscope, how nicely it can resolve here the neuronal features. So that's just as a side remark. And now let me conclude my talk. I know I'm a little late, I'm sorry. I took almost the whole hour now. So the conclusion was here, as I told you, that indeed the anatomy also seems to be consistent with the idea that you have from the PPC areas, at least, the possibilities that activity shifts either frontally or posteriorly. Now the last thing that Yazir did was that he also tried to cast all these findings from his comparison with the auditory and tactile task. He tried to cast it into like a modulation index. So he actually calculated two indices, a task index and a strategy index. So you can either ask, well, what if you look at this dorsal cortex view, what are the patterns in terms of this, if you have the same strategy but different tasks? Yeah, for example, here you really have active tries, independent whether they are in auditory or tactile task, and you have passive tries, also auditory versus tactile. This would give you a task index. Or you ask, what do they look like for the same task, but for the different strategies, active versus passive? Yeah, this would be the strategy index. And if you then color-coded here for the different phases, in the sensation phase, then it turns out that the task index is very high. So green means here it's this is what is more engaged during the auditory task, and the more bluish would mean more engaged in the tactile task here, right? But it's rather flat than here in terms of the strategy index. There's not so much difference. But in the late phase of the task, in the delay phase, when the sensory stimuli are over, then we see that the task index is not that prominent anymore, but that then the strategy index is high, just meaning that in this late phase, the activity patterns also are much more reflecting the behavior that has happened during the sensation, right? The behavioral strategy that has happened with, again, this distinction of M2 and PLA. You can also look at it as a time course. I like this, where he essentially calculated this index throughout the entire trial time, right? These are the maps just from these 2 time points. If you look at the entire trial time course, essentially the task index builds up during the pre-stimulus phase, almost like in expectation of the stimulus, and then it's highest and peaking when the touch or when the stimulus happens. But then at that time, also then what the animal does in terms of behavioral strategy matters, and more this becomes a dominant feature, what the activity pattern looks like in the delay phase, and it's then governed a lot by the behavior of the mouse. So to conclude, here from this, the distinct PPC areas are engaged depending on the sensory modality, that's during the sensory stimulus, while the strategy-dependent location of delay activity generalizes across modalities, where we really find these mediofrontal versus posterolateral areas. And this is in a way just illustrated here. You can see it here summarized for the delay phase, the shift to the M2 or ACC areas, and in the passive for the passive strategy, the shift to the posterolateral association areas. Now there's many, I think, questions that arise from these studies. Obviously, we're very interested to look more into the details of the pathway-specific interactions and the pathway-specific signaling, where we have started a little bit with the anatomy in the PPC areas, whether you really have two distinct pathways emerging from PPC areas. There's a very, I think, important question that obviously I think and expect other areas to be important and much involved, in particular thalamic areas, which are interconnected with the cortical areas here. That's something we're looking into right now. It will be interesting to look into these regional interactions and the interactions between like frontoparietal regions, the classic frontoparietal network, if you like, during real working memory. I mean, I avoided to term our task working memory because it's a short-term memory task, but not a real working memory task where you have to basically hold a stimulus and work on it only later upon the second stimulus. That's something which will be interesting to look into with the wide field imaging as well. And then the mechanisms of convergence, because obviously the mice can solve the task correctly for both of these strategies. So at some point, both of these delay patterns have to converge onto the same action, the correct licking, right? And this is something also which we still haven't found out and looked into. And then coming back to the beginning of my talk, obviously also we are highly interested to look further into the learning mechanism, into finer details of what cellular mechanisms might underlie the circuit adaptations and the sort of emergence of the correct signal flow during the task and making the animal enabling it to solve the task. So with this, I'm sorry if I was a little too long, but I hope you still have time for questions. I want to thank the two main people again, Ariel Gilad and Yasir Galero Salas. They also had help specifically for these tasks from Dominic and Balázs. But I also like to thank the whole rest of the group. This is our institute. At the moment, we have a little bit of snow here in Zurich, but in summer we sometimes obviously also like to go to the mountains and hike. And with this, I thank you very much. I hope you are still here, and I hope you have some questions.
Okay, thank you, Richhoff. That was an excellent talk. Fortunately, you had a lot of interesting things to tell us, but unfortunately, some people had to go for classes and things at 12:15, so we've lost some of the audience. But I think we have time for a couple of questions. One question was in the chat from Dieter Jaeger. I think he had to leave, so I'll ask the question on his behalf. He was asking, is the activity, particularly in the memory period, bilateral, and have you looked at bilateral activation versus unilateral activation?
Maybe I stop sharing, right? Or I don't know if I need to come back. I stop sharing for a moment. Yeah. Let me see. No, we haven't. No, we have not. The only thing we did is that for the optogenetic experiments, we used bilateral stimulation of the PPC areas because we presumed it may have a weaker effect if we don't do it bilaterally. So, but initially we always just did the preparation on one hemisphere, and we never really so far have looked at the really entire
Yeah,
cortex. So
yeah,
it's something still to do,
yeah.
definitely.
I imagine Dieter was possibly asking this because of the prior work showing the importance of the bilateral activation for the licking activity in ALM with some of the other somatosensory tests that like the Spuato group has used. So
Yes, I'm also very interested in looking into this further because also the ALM activation that like Carol sees for the left-right licking task, which is more
yeah,
like a skilled licking task, right?
yeah,
Is in a different spot, right? And my interpretation in a way is that it has to do with it. That's my hypothesis that it has to do with the difference of the go no go task, where it's more like licking versus not licking, and in his case, it's already in a way one step further because it's skilled licking to the left or to the right. Yeah,
yeah.
so it may make sense that it's already shifted to pre-motor areas that are more closely related to the skilled licking.
Yeah.
Um, mhm.
Maybe we have time for one question if someone wants to unmute and ask a question. Otherwise, I can fill in with some of the other questions in the chat.
Ah, yeah, I can now see the questions also.
Mhm. So yeah, I think Dieter, of course, had several questions.
Another question.
One was related to the differences in, I guess, the early trials that were not analyzed, um, where there was licking during the delay, were those common in the active state trials? I imagine those had a close relationship.
Ah, good question. I probably shouldn't really say I know it by heart. I would need to look at it again. I'm not sure. I cannot answer it. I'm sorry.
Okay.
I shouldn't say anything because I just don't know. I would actually look myself. As I said, in a way, we almost ignored and just excluded these trials from the analysis, even though they would be interesting to look at.
Yeah.
I may have to convince Ariel also still to go back and actually look at it and do some analysis on them. Hmm.
Yeah. Okay. So I'm sure there's plenty of other questions. I would encourage those of you in the talk to reach out to Fritjof if you have your follow-up questions. For those of you who aren't meeting him in individual meetings, but for the sake of time, so that everyone can move on with the things they need to do, we'll end it there. And thank you, Fritjof, for a really rich and excellent talk. And we'll
Thank
chat in a
you.
bit.
Yeah. Thank you.
Okay.
And see you later. Yeah,
Yes.
Pilar. Hmm.
Yep. Thank you.
Okay. Ciao. Ciao.